Hello everybody, my name is Marzieh. I'm a PhD candidate at Boston University in bioinformatics. I work in Gary Benson's lab, and today I will be talking about population-wide study of VNTRs in the human genome. By an introduction of what VNTRs are. So, minisatellites or tandem repeats are, as the name suggests, patterns of nucleotides repeated right next to each other. The mini in the minisatellites means that the pattern length is greater than 7 base pairs. A minisatellite loci is defined by its position on the reference genome, the pattern or the pattern consensus, and the copy number. Here I show you an example. It's just a region in chromosome 1. In black you can see the flanks, and the patterns are repeated in different colors: blue, orange, gray, yellow, blue, and then green. And as you can see, these repeats do not have to be exact and do not have to be complete. Variable number tandem repeats, or VNTRs, are minisatellites that have different copy numbers in a population. The allele for the VNTR loci is a copy number, and usually we express it as the amount of the copy number gain or loss compared to a reference genome. And here is an example that you can see that individual 1 has 4 copies of the VNTR and individual 2 has 3 copies, and this will become a VNTR. VNTRs are predicted to have heterozygosity between 40 to 60%. They have high mutation rate between 10 to the minus 3 to 10 to the minus 7. As a reference, the mutation rate of SNPs is 10 to the minus 9. There has been in the literature shown that they can affect chromatin organization and cause structural variations. And VNTRs that occur inside introns may affect alternative splicing, and the ones that occur the VNTRs that are inside the promoter regions can affect the expression of the genes. Important to study VNTRs. First, they play a role in evolution. A recent paper published in 2019 compared VNTRs in the primate population to human population, and they found that the gain of VNTRs can be associated with gain of cognitive abilities, such as language. They've also been associated with various diseases. Neurodegenerative diseases can be explained by VNTRs, Alzheimer's disease, Huntington disease, and many types of disorders, depression, anxiety, aggressive behavior, have been associated to different VNTR alleles. Various cancers have been associated to VNTR alleles, and longevity. Studies on centenarians in Italian populations and Danish populations have been associated to different VNTR alleles. So here I'm going to use the data from the 1000 Genomes Project, which was generated at the New York Genome Center. These genomes were sequenced at 30x coverage with read length 150 base pair. They consist of 2,504 individuals from 26 different populations and 5 superpopulations, as you can see on this map here. The longer read length allows us to genotype VNTRs, and the coverage gives us statistical power. There are no relatives in this data set. So we downloaded the TR loci on the GRCH38 reference genome. You can get these from the Tandem Repeat Database, TRDB, which was produced in our lab. There's over 190,000 TR loci, which consist of a little over 0.5% of the whole genome, which is a very small amount. But when you look at the annotations here on the right, you can see that 63% of the TRs are inside genes, 12% are in transcription factor binding sites, and 2% are, if they're not inside genes or transcription factor binding sites, they are in regions that overlap with open DNAs. Interestingly, about half of the genes contain at least one tandem repeat, and 2,990 TRs overlap with coding exons, which is very significant, and 5% of all transcription factor binding sites span TRs. All this suggests that these VNTRs are not random positions in the genome and probably have a regulatory function. So here's the methodology. I'll just go over it, and I'm going to present my results in 3 different parts. First, we're going to genotype VNTRs using VNTR-seq, a tool that was made in our lab again. And we're going to define common VNTRs. We're going to then use some machine learning and do some population-wide analysis of these common VNTRs. And finally, I'm going to investigate the potential effect of VNTRs in gene expression. The first observation we had is that these VNTRs, the number of VNTRs per sample, depends on the evolutionary distance of the individual to the reference genome. Here you can see a plot. On the x-axis, we have the read coverage, which is the number of base pairs in the dataset, the reads, divided by the genome length. And on the y-axis is the number of VNTRs we genotype for that individual. And you can see for the same read coverage, we find more VNTRs for African people with African ancestry, which is the red line here. And you can see we find the least for European ancestry. This is expected as the reference genome consists of mainly European ancestry. Second thing we looked at is heterozygosity. So we looked at the ratio of heterozygous VNTRs over the total number of VNTRs. The reason we didn't calculate the zygosity is that here we do not have finite number of alleles. We can have any number of alleles. We can gain copies and copies, and it's just going to be an integer number that goes up. As you can see, the Africans have the highest heterozygosity, and the East Asians have the lowest. And when you look at that compared to the numbers that were predicted in the literature, they are very similar. Also, I compare them to heterozygosity of SNPs that was calculated in 2009 on the same populations, and the results agree, with Africans having the highest and East Asians having the lowest. Our Americans do not agree, mainly because our populations do not correspond to each other. Our Americans had a lot of Puerto Ricans, which clustered with the Europeans mainly, and we had a Colombian cluster that is actually this long tail down here. So we identified common VNTRs as TR loci that were genotyped as a VNTR in at least 5% of the individuals, that is, at least 126 people. So we use this terminology to make a comparison to SNPs, common SNPs, but here we do not have finite alleles, as I said. There are total 5,699 common VNTRs with 18,253 alleles. These alleles, just as a reminder, are the copy number of that VNTR in that individual. And from now on, I will just, whenever I say VNTR, I mean a common VNTR. So we did some enrichment analysis on the VNTR loci. So looking just at the annotation of them, out of these 5,699 common VNTRs, they overlap with over 4,000 genes and over 650 exons and 300 UTRs, which is pretty significant. It shows that majority of these common VNTRs are not intergenic or like junk DNA, but actually occur inside genes. We did a GSCA gene set enrichment analysis on KEGG pathways for these 650 exons, and we found pathways related to heart and neuron function and cancer. On the right, I'm showing you genomic loci overlap enrichment, which we did using LolaWeb. It's a tool that was published in 2018. And you can see that when looking at the ENCODE segmentation enrichment, our VNTR loci are enriched inside enhancers and weak enhancers, and also CTCF segments. So all of these have p-value less than 1%, and the odd ratio is greater than 1. And CTCF segments, as you know, are responsible for chromatin organization. We performed unsupervised clustering on our common VNTRs. We selected the top VNTR alleles that, by variation, and we saw that the different populations can be easily separated using the principal components, where the first principal component separates the African population, which is represented in red and orange here. The second principal component can separate the East Asians, the third South Asians, and the fourth the Americas. As you can see, each PC captures very little variation, and that is because the variation inside each cluster, each population, superpopulation, is still very high. So then, using these principal components, we tried to predict the ancestry. I use first 10 principal components, and I learned a decision tree. I did not need to do a random forest because the decision tree gave good enough results. And looking at the data, that was pretty balanced for the different populations at about 20% in each population. We could achieve very high accuracy, precision, and recall. This is a method that is used for using SNPs in 23andMe and DNA Ancestry, and you can see that we could achieve similar precision using just 10 principal components and only 4,000 loci, which is pretty significant. So seeing that I can separate all these populations using these different alleles, I was wondering if I can find VNTR alleles that are population specific. So I did a Fisher-Exact test to find VNTR alleles that are overrepresented in each population. And here I'm showing you a Venn diagram of the number of VNTR loci that have an allele specific to that population. As you can see, the Africans here with 230 have the highest number of VNTR loci, which is expected as they are more distant, followed by the East Asians, which have a 63. But there's a good number of VNTR loci that overlap between these different populations, 56 of them, suggesting that these 56 loci have alleles specific to each one, making them hyper VNTRs. So we did some validation in the lab, and I got these gels that we ran for our validations, and I was wondering if I could do in silico presentation of these VNTRs for the different populations. So we made this vitral gel presentation. On the x-axis, we have the VNTR loci, and on the y-axis, I have the different VNTR alleles, which are the minus numbers are losses, so loss of 1 copy, loss of 2 copy, and the positive numbers are gain, all compared to the reference genome. And for each population, African, American, East Asian, European, and South Asian, I draw bands which the density represents the number of people from that population that had that allele, and the Y is showing the allele that it is. And you can see that some of these alleles are specific to that population. For example, if we look at the first loci on the left, you can see that the Americans have a significant loss of 1, which other populations do not observe at the same rate. In the squares, you can see the different alleles are specific to different populations. We'll present a Shiny app that people can browse and make these vitral gels. They can look for different genes. As I said, half of the genes have VNTR loci in them, and they can extract data as they wish. Looking at the significant number over 5,700 common VNTRs, which 1,200 are population specific. The next thing I want to investigate is that are these VNTRs affecting the gene expression? To do so, I did a chi-square test on the genotype and the expression if a VNTR was inside the intron of the gene in an exon, if it overlapped the 5' UTR or the 3' UTR, or it was in the promoter region, which is 1,000 base pair upstream. So I'm just showing you 2 examples here because I found hundreds of genes to be significant. I had 800 individuals from the same population. These are all whole blood mRNA data. And as you can see here, for example, I'm showing you one type of association that I see. This is the IRF5 gene, and this is a protein, the isoform D. IRF5 is related to immune system response and viral infections. And as you can see, when you have the 0 allele, you have a higher expression for this protein. So people who have a homozygous loss of 2 copies have significantly lower expression than people who have an allele that is the same as a reference. And you can see that this is a linear increasing. But there's another class of effect of VNTRs on the gene that, for example, here you can see that in this case, if you do not have the 0 allele, which is the same as a reference, you have some of the people switching on and having a very high, or in some cases very low, expression of that gene. Here you can see that I'm showing you this NA class, and the reason to show that is that the fact that we observed 138 00s, 01s, 11s, NA, although we didn't observe any allele for those individuals for 246, but we know that they're not going to be 00, 01, or 11 because we could detect those, and we know these are something that could not be detected. So that's why I'm showing them as a class for themselves. But here you can see that in the absence of a 0 allele, we get some individuals with a higher probability are going to have very high expression. So to conclude, this is the first population-wide study using Minisatellites in the human population. We found 5,699 common VNTRs, which overlap with over 4,000 genes and 650 exons. A good proportion of them, over 1,200 of these common VNTRs, are population-specific. We can predict populations with high accuracy using VNTR alleles. And we also showed that VNTRs that overlap with introns and exons or are inside the promoter regions may affect splicing and expression of genes. And this work was funded by the NSF grant IS 1423022. Thank you everyone for listening.